We'll just start in a couple of minutes. Right, so I'll start sharing my slides. Right, welcome to the 11th live session of structural biology. So today we'll be talking all about protein engineering. So what is protein engineering? Now we have learned earlier that proteins are large macromolecules that are composed of one or more long chains of amino acid residues. Proteins differ from one another primarily in their sequence of amino acids, which is dictated by the nucleotide sequence of their genes and which usually results in protein folding into a specific 3D structure that determines its activity. So what this means is that DNA is made up of different base pairs, and through a process of transcription from DNA you can get RNA, and using a process called translation from RNA you can get protein, and each triplet of base pairs codes for one amino acid in the protein. So if the base pair sequence changes, the amino acid also changes, and you can get different sequences of amino acids with different triplet base pairs. And so improvement in the activity and usefulness of an existing protein or creation of a new protein function can be done by making suitable changes in the amino acid sequence. And this is called protein engineering. So as you know that depending on the sequence of a protein, it can adopt different structures. And depending on the structure, it can have different functions. So if we want a specific function of the protein, we needed to have a specific sequence. And we can modify that sequence by changing the base pairs in the parent DNA. Now, protein engineering utilizes recombinant DNA technique to introduce the desired changes in amino acid sequence of enzymes. In addition, increase in the level of production of protein is also part of protein engineering. So if you remember the concept of recombinant DNA, basically you take a bacteria, which in this case is Agrobacterium tumefaciens, and you take out its plasmid. Then using restriction enzymes, that part of the plasmid is cut out, in place of which you want to introduce the desired gene. So once that part is cut out, you have DNA containing the gene of interest, and using DNA ligase, you can attach it to the part of the plasmid that was cut out. Next, the plasmid is reinserted into the bacterial cell and allowed to multiply. So like that, you can get the new organism with your desired trait originating from the new gene that has been inserted. So this is the recombinant DNA technology, and using this, a desired protein can be manufactured. Protein engineering is a relatively young discipline, with much research taking place into the understanding of protein folding and recognition for protein design principles. So protein engineering is still in its very basic stages, but we'll go into that in the subsequent slides. So what is the protein architecture? We have learned about this before, but I'll still go into it in short. So as you know, amino acids are the building blocks of proteins, and they consist of a carboxyl group, an amino group, and a side chain, and an alpha hydrogen. So the side chain can be of 20 different types, giving rise to the 20 natural amino acids. Now, two amino acids can undergo a dehydration reaction to form a peptide bond. So the COOH group of one amino acid will lose the OH group, and the NH2 group of the other amino acid will lose one of its H. So together an H2O molecule is removed, and so it's called a dehydration reaction, and a peptide bond is formed, which is basically a C double bond O NH bond, and the water is released. So through this peptide bond formation, many amino acids can be joined together to form a secondary structure. So secondary structure is basically formed from a chain of amino acids which are joined together by peptide bonds, and it adopts structures like an alpha helix or a beta sheet due to intermolecular hydrogen bonds. So secondary structure is the result of hydrogen bonding. From secondary structure, a tertiary structure can be formed, which is basically a three-dimensional folding pattern due to side chain interactions. So you can see you have two types of secondary structures here. This blue one, which is the alpha helix, and the red, which is the beta sheet. So the secondary structures come together through folding and are held together by different side chain interactions, forming a tertiary structure. The final level of organization is when two or more tertiary structures come together to form the quaternary protein structure. So here you can see there are two tertiary structures forming a dimer complex. So that is the quaternary structure. Now you can have other types of tertiary structures called supersecondary structures. Like if you have a bundle of 4 helices denoted by 4 alpha signs, then that is called a 4 helical bundle. Or you can have alternating beta sheet, alpha helix, beta sheet, alpha helix, beta sheet. And that would be a Rossmann fold. So different secondary structures can be arranged among themselves to form a supersecondary structure. Next, we'll quickly go into kind of a revision of the different torsional angles and the Ramachandran map. So if you remember, this is showing different amino acids held together by peptide bonds. So this is the peptide plane. Since C double bond O NH has partial double bond character, so the peptide groups lies in a plane. Now, for the phi angle, you begin at one of the carbonyl carbons, then go to the adjacent nitrogen, C alpha, and the next carbonyl carbon. So the dihedral angle between these 4 atoms, starting from one carbonyl carbon to the next carbonyl carbon, is known as phi. The psi dihedral angle is when you start from a nitrogen, go to alpha carbonyl carbon and to the next nitrogen. So the dihedral angle between these 4 atoms is known as chi, from one nitrogen to the next nitrogen. Now the dihedral angle omega, you start at one C alpha and you go to the next C alpha. So starting from one C alpha, that is CO, NH, and another C alpha. So this is the dihedral angle omega. Now omega can have 2 values. One is near 180 degree when it is called S trans. So here you can see the dihedral angle between one C alpha and the next C alpha is close to 180 degree. And so you are calling it an S trans orientation. But if the omega dihedral angle is near 0 degree, as is shown in this example, then you call it an S cis orientation. And finally, we have the chi dihedral angles. So chi is basically the side chain dihedral angles. So suppose you have a lysine side chain that is shown here. You start from the nitrogen 1, 2, 3, 4. So that will be one of the chi angles. And as you go through the different atoms of the side chains, you can have chi 1, chi 2, chi 3, chi 4, different dihedral angles. So now that you have looked at the 4 kinds of dihedral angles, now we can focus on the Ramachandran map. So some combinations of the torsional angles are much more likely to occur than others. So these angles which are likely to occur fall on the allowed regions of the Ramachandran plot. So Ramachandran plot basically has the psi angle in the y-axis and the phi angle in the x-axis. Now the white regions are the disallowed phi psi, meaning that in proteins or peptides, these you do not find these phi psi angles because they are sterically not allowed. The yellow region, the red regions are the allowed phi psi angles, and they constitute the beta sheet, which is in the top left part, and the alpha helix, which is in the bottom left part. The yellow regions are the border space, meaning that they are not as much allowed as the red regions, but they are still sterically allowed and not disallowed as the white regions. So within this border space, you can find the left-handed alpha helix in the top bottom side, this yellow region that is there. So Ramachandran map basically gives you an idea. If a predicted, suppose you have a predicted protein from a software and you want to find out the chances of that protein occurring in reality. So if the phi psi angles of that protein are plotted and most of them fall in the allowed regions or the border space, then you can say that yes, it is a correctly predicted structures. But if it falls in the disallowed regions, that means the structure needs to be improved because phi psi angles in those regions are not sterically allowed. Next, we will look into the relation between evolution and proteins. So what is evolution? There are many factors which are responsible for adaptation, optimization, and innovation in the living world of proteins or enzymes. So all these factors which help in adapting, optimizing proteins so that they can best thrive in the correct environment is called evolution. Evolution executes a simple algorithm of diversification and natural selection, an algorithm that works at all levels of complexity from single protein molecule to whole ecosystems. So what does that mean? Suppose you have a protein the original parent protein, which has a stability delta G of -10. So the more negative this value, the more stable the protein. So the parent protein have the stability of -10 kilocalorie per mole. Now suppose in the course of evolution, there is a destabilizing mutation. Like maybe one of the alanines in the protein has been changed to threonine or some other amino acid. So that mutation is causing the energy to rise to say around -7 kilocalorie per mole. So the mutation is still allowed because it falls within the stability threshold. So if the stability threshold is -5 kilocalorie per mole, the mutation is still allowed because you have the mutation energy which is below the threshold. Now, if the parents in this new protein have -7 kilocalorie per mole, so these are the parent. So when this new protein is multiplying, it will give rise to proteins of -7 kilocalorie per mole stability. But now if you have another mutation, it will cross this threshold because it will go, it has gone to say -4 kilocalorie per mole. Now this second mutation is not allowed because it is less stable than the allowed threshold. So this mutant, the second mutant protein is not fulfilling the stability threshold. So organisms with that protein will start to die, and this evolution will not be allowed. So only that evolution is existing, which is allowed to exist due to stability reasons. So evolution helps in improving the protein and not in uh, making it less stable. Another example has been shown here. So if you have an unfolded protein chain and you have a mutation, so this mutation can result in several things. Firstly, because of the mutation, the protein is not folding correctly. Instead, it is aggregating among itself. So that mutation will not be allowed. Or another thing can happen is that because of the mutation, it does not bind to the original ligand, but it will bind to a new ligand. So that protein can be used for some other purpose. But if this new interaction that is there is binding and causing a negative function, then that evolution will again not be allowed. Another thing can happen is that the new mutation has no effect. That is, it is neutral and it is still binding to the original ligand. So it will continue on as it has. So mutations can have negative effect, positive effect, or no effect at all. So that is how, like if it has a negative effect, that mutation will be discarded by evolution. If it has a positive effect, like it is binding to a new partner and that is giving some positive effect, then that mutation will be allowed, and slowly the original protein will start to die out and the new protein will dominate. Or it can be a neutral mutation where it's the structure doesn't change and it still binds to its old partner. Now we'll just revise what we have learned in the first class, which is polymerase chain reaction and Sanger sequencing. So in polymerase chain reaction, there are three main steps. Firstly, the denaturation, which is separation of two strands of DNA molecule. So if you have a DNA molecule and it is heated to 95 degrees Celsius, this will break the hydrogen bonds between the base pairs and the two DNA strands will separate out. Each strand can now act as a template on which the new strand is built. So how this happens is that the temperature it is called annealing. The temperature is reduced to around 55 degrees Celsius so that primers can anneal to the template. So primers are small sequences of DNA that attach to the DNA template at the beginning so that it can start off the process of DNA replication. So annealing is needed, the annealing of the primer is needed because the DNA polymerase can only add a nucleotide when there is an existing 3' OH group. So the initial annealing is needed to give the existing 3' OH. Once the initial primer has been annealed, the temperature is raised to about 72 degrees Celsius and DNA polymerase starts adding nucleotides one by one at the end of the annealed sequence. So that helps in forming the entire extended DNA. And from one copy, you can get two copies. And similarly, from 2, you can get 4, 4, you can get 8. And after n cycles, you will have 2 to the power n copies. So polymerase chain reaction rapidly gives, like if you have a very small DNA sample, you can carry out polymerase chain reaction to rapidly get a large amount of that sample. Next, what is Sanger sequencing? So in Sanger sequencing, it is a special kind of polymerase chain reaction where the user mixes a low ratio of chain terminating dideoxyribonucleotides or DDNTPs with the normal deoxyribonucleotides DNPs in the polymerase chain reaction. So you can see this is the normal deoxyribonucleotide or DNTP, And this is a dideoxyribonucleotide or DDNTP, which does not have the 3' OH group. So this OH group at the 3' position is not present. So because of this, this OH group is necessary for phosphodiester bond formation in order to extend the DNA. So due to absence of this group, it results in chain termination. So whenever during the polymerase chain reaction, instead of a DNTP, a DDNTP is incorporated, the extension of the DNA chain stops. So what happens is we get a lot of oligonucleotides terminated at different lengths by the DDNTPs. So instead of the full sequence of the DNA, suppose we can get a tetramer, then an octamer or a heptamer. At different lengths of the DNA, we will get terminated at different lengths due to the DDNTP. Then we can separate these by size using capillary gel electrophoresis. So allowing the fragments of the DNA to move through a gel with charged separation. So the larger fragments will move slowly and will remain at the initial positions, while the smaller fragments will move fast and go downwards along the gel. And then using a laser beam, we can detect the different sequences, and using that, we can sequence the DNA. Using the laser, we'll get an idea of the what are the sequences of different lengths. So we can put together the puzzle like a jigsaw puzzle is solved. So if we know what is the sequence of the dimer, trimer, tetramer, heptamer, we can put that together and get the entire DNA sequence. So that is called Sanger sequence. So what can be engineered in proteins? Now we can engineer the protein folding or structure. So, there is a thermodynamic equilibrium between the native folded protein and the unfolded state. So, suppose this from unfolded state, it goes to folded state. It goes through several intermediates. And there you have heard of the molten globule model, where initially there is a large hydrophobic collapse of the protein, where from an unfolded state, the side chains come together forming hydrophobic interactions to form a molten globule state, which is kind of an intermediate state. And then the different secondary structures form by hydrogen bond formation to give rise to the native state of the protein. Now, if we can somehow control the folding process, we can get a different protein structure. And different structure will have a different function. So like that, we can control the structure of a protein. And this can be done by targeting its thermal or environmental stability, or its stability in different pH, solvents, salts. So if we target its stability in different conditions, then it will form a different structure in order to gain the maximum stability at that particular condition. Also, the function of a protein can be controlled, like its binding interaction with its surroundings. So if there is a protein and we can rationally design, we can rationally design a protein such that it will bind to our target of choice. And we can also carry out random mutagenesis or mutate different residues of the protein in order that it binds to the target of our choice. So using these two different methods, either rational design, by which we computationally predict that what should be the amino acids at the active site in order to best bind to the target of choice, and then carry out that synthesis. Or we can use random method in which in the original sequence of the protein, the amino acids are randomly varied and computationally screened to find the one that will best bind to the target of the choice. So like that, we can control the function of the protein. Or the catalysis can also be controlled, that is the increased binding to the transition state. So if you have an enzyme, what happens is that it will bind to the substrate and it will form the enzyme product complex, and the product will be released and give rise to the original enzyme, which can again be reused. So if we have a knowledge of the catalytic mechanism, that is which amino acids are contributing to the catalysis and how they are binding to the protein. If we have a knowledge of that mechanism, then we can easily engineer a certain combination of amino acids such that the binding to the transition state is maximum, and that will also increase catalytic rates. So like that, the function of a protein can also be controlled. Now, there are several factors which contribute to the stability of the proteins. One is hydrophobicity. So, there are various hydrophobic amino acids like leucine, valine, or isoleucine, which has a hydrophobic side chains. So, there can be van der Waals interactions between their side chains, and that is called hydrophobic interactions. Or there can be electrostatic interactions. So if you have a lysine, which is a basic amino acid, it will have an NH3 plus, and an aspartic acid, which is an acetic amino acid and will have a COO minus, then these two can have an electrostatic interaction, also called a salt bridge. Or there can be hydrogen bonds. So in alpha helix or beta sheet, you know that there is in alpha helix, there is 4-to-1 hydrogen bonding. And in beta sheet, there is hydrogen bonds between the adjacent beta strands. So hydrogen bonds also contribute to the stability of proteins. Then there can be disulfide linkages. So two cysteine residues can come together and both will lose the hydrogen from the SH to form a disulfide bond. Or there can be metal chelation. So if you have, say, an acidic amino acid with COO minus, from both sides, it can coordinate to a positively charged metal ion, and that will also help in holding the shape of the protein. Another way is the reduction of the unfolded state entropy with X to pro mutations. So basically, the unfolded state has a large entropy and the folded state has lesser entropy. So that is one factor that can bias the protein to go from folded to unfolded. But if we incorporate a proline mutation in the unfolded state, then the entropy of the unfolded state will reduce, because proline is a cyclic amino acid and it is not free to rotate. So if the entropy of the unfolded state is reduced, then folding becomes easier, and it also contributes to the tendency of the protein to go from unfolded to folded state. Another can be the stabilization of alpha helix dipoles. So you know that the C terminal of alpha helix is negatively charged. So if you add a basic amino acid like arginine, which will have an NH3 plus, it will stabilize the negative charge of the C terminal. Or the N terminal is positively charged. So if you have an acetic amino acid like aspartic acid, then the negatively charged aspartic acid COO minus can stabilize the positively charged N terminal. This is called N or C capping. Or there can be salt bridges between basic and acidic amino acids like glutamine and lysine, which is stabilizing the alpha helix. So there are several factors which contribute to the stability of proteins. Just give me 2 seconds. Right. So the first strategy of protein engineering is called rational protein design. Rational protein design approaches make protein sequence predictions that will fold to specific structures. These predicted sequence can then be validated experimentally through methods such as peptide synthesis, site-directed mutagenesis, or artificial gene synthesis. So what does this mean? Initially, in rational protein design, there is computer-aided design, meaning that using statistical and structural parameters, the computer can design that what is the protein structure that will enable the function that we want. So initial step is using the knowledge of pre-existing protein structures and functions, using that pre-existing knowledge, using the computer to make predict a design for our engineered protein. The next step is to change one or more nucleic acids in the plasmid to change the amino acid in the protein. So as we saw before, from DNA you get RNA, and from RNA you get protein. So if we change one or more nucleotides in the DNA, we will get a different protein. So based on the rational design of the protein that we have, we can change different nucleotides in the original DNA in the plasmid to get our desired amino acid in the new protein. Then the plasmid is injected into the bacterial cell. So the bacterial cell is altered by introduction of this engineered plasmid, and it is allowed to replicate. Once that it has replicated, the protein, the bacterial cell is expressed. So a large amount of the protein is obtained. Then the cell can be lysed and removed, and the protein can be isolated. Then the protein can be purified. So if you do lysis of the bacterial cell, you will get a mixture of several proteins. So we can separate the target protein by size exclusion or ionic chromatography, and we get the target protein. Then the biochemical testing can be done to find if the target protein indeed has the desired function for which we are doing the protein engineering. Then we can, based on the results of the target protein, we can do even more rational design to get a better structure. So this is a kind of repetitive process. In general, this has the advantage of being inexpensive and technically easy, since site-directed mutagenesis methods are well developed. However, the major drawback is that detailed structural knowledge of a protein is often unavailable. And even when it is available, it can be very difficult to predict the effects of various mutations, since structural information must provide a static picture of a protein structure. So what this means is that, as I mentioned, for the rational design to be done, we need to have a very detailed knowledge of the protein structure and function. And many times this is not available because the structural information that we get from, say, X-ray crystallography or cryo-electron microscopy or NMR gives us the picture of the static protein. That is when it is not moving. But all this binding and interaction studies happens when the protein is dynamic. So since we often do not have that dynamic information, the performing the rational design becomes difficult. Now, there are several steps in the rational design, which is firstly to have a target structure. The protein function is heavily dependent on protein structure, and rational protein design uses this relationship to design a function by designing proteins to have a particular structure or form. So, like we discussed before, from a protein sequence, you get a specific structure, and it is the structure that determines the function. So if we want a specific function, we can design that structure accordingly. But for this, a detailed information of structure function relationships is essential. So by definition, in rational protein design, the target structure or ensemble of structures must be known beforehand. Most often, the target structure is based on a known structure of another protein. That is why that structure function relationship is very important. Just give me 1 minute. Right. The second step that is known is the sequence space. So in rational protein design, proteins can be redesigned from the sequence and structure of a known protein or completely from scratch, which is known as de novo design. In protein redesign, most of the residues in the sequence are maintained as their wild type amino acid, while a few are allowed to mutate. In de novo design, the entire sequence is designed anew based on no prior sequence. So there are two ways of this rational protein design. In first, you identify a few amino acids which are contributing the most to the ligand interaction or the function of the protein, and you mutate those particular amino acids only, and the rest of the sequence is kept same. The other method, called de novo design, is basically designing the entire protein sequence from scratch. So that takes a lot more time. The next point is the energy function. So rational protein design techniques must be able to discriminate sequences that will be stable under the target fold from those that would prefer other low energy competing steps. Thus, protein design requires accurate energy functions that can rank and score sequences by how well they will fold to the target structure. At the same time, however, these energy functions must consider the computational challenges behind protein design. So basically, suppose you have an isoleucine residue. There is a certain backbone and side chain conformation that it will prefer. So using that information of the backbone and side chain conformation that is preferred, it is possible to predict how the protein will fold if a certain sequence is known. So there are computation programs called energy functions, in which if you input a particular sequence, the program can predict that which is the structure that it will fold to. That is, which is the energetically most stable structure allowed for that sequence. So these computer design strategies can screen these sequences and predict that which are the sequence that will fold to the structure that we want, because only that particular structure will give the function that we want. One of the most challenging requirements for the successful design by this energy function is both accurate and simple computational calculations. The problem is that if we want a very accurate prediction, the simulations become too slow and impractical for design. But if we have a fast simulation, then the results are not that accurate. So that is the most challenging problem. Molecular mechanics force field, which have been used mostly in molecular dynamics simulations, are optimized for the simulation of single sequences. But protein design searches through many conformations of many sequences. Thus, molecular mechanics force field must be tailored for protein design. So, like I was saying, you can have coarse-grained function which compute very fast but are not that accurate. So if you can see, the predicted energy minima are quite broad. So they are predicting several different structures and not just one structure. But this is a very fast method. Or you can have atomistic function, which is more sensitive to structural detail. So as you can see, the energy minima are very sharp. So it is predicting like only a single structure or a couple of structures. But so although this is more accurate, it computes very slowly. So you can either have an accurate result or a fast result. There are several challenges for effective design function. Now, as you know, water makes up most of the molecules surrounding protein and is the main driver of protein structure. Thus, modeling the interaction between water and protein is vital in protein design. So suppose you have a random protein, then definitely there are structures like hydrogen bonds or hydrophobic interactions contributing to the stability of the protein, but water is also an important factor. For example, it is because the water molecules do not want to surround the hydrophobic groups. Rather, they want to form hydrogen bonds with other water molecules, that this unfolded protein is forming this particular orientation. So it is necessary for effective energy design functions to have different, to account for the presence of water molecules. Now, the number of water molecules that interact with a protein at any given time is huge, and each one has a large number of degrees of freedoms and interaction partners. So the problem is that if we have to make a model which has that many number of water molecules as is present in practical proteins, that becomes very difficult. So that kind of water models is called an explicit solvation model, where each water molecule is modeled separately. But since this is very difficult and it takes a long time, most protein design programs model the water as a continuum, modeling both the hydrophobic and solvation polarization. So that is called an implicit solvation model, where instead of considering each water molecule, the entire water system of molecules is modeled as a continuum having the properties, average properties of all the water molecules. The individual water molecules can sometimes have a crucial structural role in the core of proteins and in protein-protein or protein-ligand interactions. So sometimes water molecules have individual roles that you cannot just make into a continuum in the implicit solvation model. So failing to model such individual water molecules can result in misprediction of the optimal sequence of a protein-protein interface. As an alternative, those particular water molecules can be added to rotamers to have a microsolvation. So in microsolvation, most of the water is modeled as a continuum, as in the implicit solvation model. But the few particular water molecules that have a specific function, say in the protein-protein or protein-ligand interactions, are modeled separately as individual molecules. So this kind of dual model is called microsolvation. Now, another challenge for effective design of the energy functions is that the number of candidate protein sequences grows exponentially with the number of protein residues. For example, there are 20 to the power 100 protein sequences of length 100. That is, if you have 100 amino acids, there are 20 to the power 100 possible sequences of those amino acids, because there are 20 kinds of amino acids. Furthermore, even if amino acid side chain conformations are limited to a few rotamers, this results in an exponential number of conformations for each sequence. Now, if a designer is interested in only the best sequence, the protein design algorithm only requires the lowest energy conformation of the lowest energy sequence. In this case, the amino acid identity of each rotamer can be ignored, and all rotamers belonging to different amino acids can be treated the same. So basically, if you consider R1 to be a rotamer at position I in the protein chain, and ER1 as the potential energy between the internal atoms of the rotamer. Next, you consider ERI, RJ to be the potential energy between atom at Ri and Rj at residue position J. Then we will define the energy optimization problem as a summation of the energy of the residue Ri. So Ri is one residue at position I. So the energy of the residue due to the interaction with its own atoms, and Eij to be the energy of residue at Ri with the residue at Rj. Now, if this is added up among all I atoms, so the energy of the atom within itself and with other residues, with all other residues except itself. So that being the total energy, if we can find the minimum of that energy, then the sequence and structure that corresponds to that minimum energy is the one that we want. So energy design functions basically follow this algorithm and give the output of the sequence and structure having the minimum of this energy. So what are the different algorithms used for this energy minimization? Several algorithms have been developed specifically for the protein design problem. These algorithms can be divided into two broad classes. One is called an exact algorithm, such as dead-end elimination, that lack runtime guarantees but guarantee the quality of the solution. What this means is that it will give a very accurate solution, but it will take time for the computation to happen. So what this dead-end elimination does is that it will eliminate clusters of rotamers, keeping the global minimum energy conformation constant. So given a residue I, a residue I1 is eliminated if the minimum energy it can obtain by interaction with the other rotamers is higher than the maximum possible energy of the initial rotamer. So what this means is that it will always keep the global minimum energy conformation, and it will keep eliminating other rotamers which have energy higher than the energy of that global minimum energy conformations. So doing that, it will guarantee an accurate result that we will always get the minimum energy conformation, but it will take time because it needs to screen through all the other possible rotamers in order to eliminate them. So this dead-end elimination is an exact algorithm, and it guarantees that the optimization process produces the optimal result for the protein design. So if there is any problem in the result of the experimental algorithm, then this means that either there is a problem in the energy function or the sequence space or the target structure, and that will need to be corrected. The second kind of algorithm is called a heuristic algorithm, and an example of that is Monte Carlo. That these can compute the results very fast, but they have no guarantee on the optimality or accuracy of the results. So this is an example of Monte Carlo, and what it does is that it randomly moves. So suppose the position is here, the initial structure that you have is here. Now there is a high probability, suppose the starting structure is here. Now that if you move to the left, there is a very low probability of 0.03 that you will get a more stable structure. So instead, the function will move to the right, where there is a high probability of 0.5a to get a lowered energy structure. So now this probability indicates that there is a lower energy structure after it, but it is not a global minimum. Instead, it is a local minimum because the probability is not that high. Now, from this, you always have a probability. You have a probability of 1 to go to the next structure. That means you are moving to the direction of the global minimum. So like this, based on probabilities, the Monte Carlo function will jump till it reaches a minimum. So because it is jumping across different conformations, it does not take that much time to compute because it is skipping a lot of conformations. But again, this does not guarantee the accuracy of the results because sometimes the function gets stuck in a local minimum instead of reaching the global minimum. There are different modifications to the Monte Carlo, like the Rosetta design, which incorporates sophisticated energy terms like backbone flexibility, or Osprey's algorithm, which incorporates the backbone and sidechain movements. So these are the two main algorithms, the exact algorithm and the heuristic algorithm, that are used in energy minimization functions. So now we will talk about mutagenesis. So as you know, 3 nucleotides can code for a particular amino acid. Now, if there is a point mutation, what this means is that there is a change in one particular nucleotide. Now, a point mutation can be a substitution, meaning one particular nucleotide is substituted with another. So instead of A, you have a C. Or it can be an insertion, that is, an additional nucleotide is inserted in the existing sequence, or a deletion, where a nucleotide in the existing sequence is deleted. So what can happen from this is that suppose you initially had AAT. If now that is, so initially you had AAT, which was coding for leucine. So if you, yes, instead of U, if you consider T over here, then AAT would code for leucine. But if it suddenly changes to another mutation where it does not code for any amino acid, that will be called a missense mutation. Like that, there can be, so a case of, suppose you have a case of silent mutation. What does that mean? So you initially had AAG, which was coding for lysine. So the G is suddenly mutated to an A. Now AAA also codes for lysine. So even though there is a mutation, the sequence of the resultant amino acids remains the same. So that means the mutation has no visible effect, and so it is called a silent mutation. Or what can happen here is that there can be mutations which have effect. Like if you had a mutation coding for a particular amino acid and changing of the sequence is coding for a different amino acid, then that is not a silent mutation, and you will get a different structure in the final protein that is produced. So what is the general strategy for directed mutagenesis? There is a hypothesis that natural sequence can be modified to improve a certain function of the protein. But the sequence must change without disruption of the structure, otherwise it will not fold to the structure that we want. So the new sequence cannot be very much different from the original sequence, otherwise the structure as well as the function will be lost. So the result is that introduction of point mutations might lead to new functions because it is changing only a few amino acids at certain points. Now that is directed mutagenesis, where only those few amino acids will be changed. Now it can be a site-directed mutagenesis, where point mutations are occurring in only a particular known area. So what happens is that you take the plasmid vector, and from that the single strand DNA template is produced. Next, the mutagenic oligonucleotide containing our gene of interest is annealed to the single strand template. Then using DNA polymerase ligase, the rest of the template is completed. So the complementary strand is synthesized so that the recombinant plasmid is produced containing the gene of interest. Then it is reinserted into the E. coli. And now when the E. coli replicates, some of the E. coli which has the recombinant DNA will produce the new protein, while the E. coli which had the original DNA will produce the original protein. So like this, a library can be created. Another way is the random mutagenesis, where point mutations occurred within all areas of the DNA of interest. So random mutagenesis produces a much larger library, because in site-directed mutagenesis, you are only carrying out point mutations in a particular area, while random mutagenesis carries out point mutation in all area within DNA of interest. So a much larger library for screening is created in random mutagenesis. It is used for investigating the structural and biological activity of DNA, RNA, and protein molecules, and especially for protein engineering. Directed mutagenesis is one of the most important laboratory techniques for creating DNA libraries by introducing mutations into DNA sequences. So now that we have a library of mutated DNA, we can easily screen through the different mutants to find out which has the optimum function that we need. There is another method called completely novel protein design or de novo protein design. Now, we had learned that in rational design, a very detailed information of the structure function correlationship is required. But in de novo protein design, no prior information is required. The protein can be designed ab initio on a computer, taking the help of protein structure prediction algorithms. So although this is much harder than conventional protein engineering, there are several advantages of de novo enzymes. Firstly, natural proteins are difficult to modify without disrupting their overall structure. But by making the protein from scratch, we can design the structure and function that we want. Secondly, enzymes with activities unknown to nature can be built using cofactors and amino acids that are not part of the standard toolkit. That is, unnatural amino acids can be used, or different cofactors that are not naturally found. So that gives us a much more wider range of functions than are available in nature if we are making the protein from scratch. The problem is that de novo protein engineering often requires weeks of computational time and iteration. So because we are making it from scratch, the time that it takes for the computer to predict is much longer than in rational design, where the computer is predicting based on a wealth of information available on the structure function relationship. So there are advances and advancements in this de novo design that is making the process more accessible, but it is still a much longer process than rational design. So in de novo design, what happens is that a hotspot is identified from the structure's target structure with the known ligand. Then several short peptides with various folds are known, and the structure or function that we want is synthesized based on these short peptides, short peptide libraries. The essential structural elements are identified, and a completely new protein is synthesized from scratch. So after that, once that new protein is predicted, it can be synthesized and then purified and used for in vivo studies to figure out whether the predicted de novo protein has the desired function. Now we'll look into protein structure prediction methods. One is called the knowledge-based method. So protein fold recognition technique involves incrementally replacing the sequence of a known protein structure with a query sequence of unknown structure. The process is repeated against all known 3D structures until an optimal fit is found. So in knowledge-based method, there are two kinds. One is homology modeling. So you have a template sequence and you have a template structure. So based on that, a 3D model can be built and then it can eventually be refined to get a better and better fit between the template structure and the structure that we want. Or there can be a process called protein threading or fold recognition, where you have a target sequence and you have a template structure library. Next, the sequence and the structure is matched such that you get the sequence corresponding to the structure that we want, and the sequence that is best fitting to the structure that we want is the predicted structure. The other method is the ab initio method. So the basic idea is to build empirical function that simulates real physical forces and potentials of chemical contacts. If we will have the perfect function and we will be able to scan all the possible conformations, then we will be able to detect the correct fold. So ab initio methods is basically like you have the unfolded protein and you are applying molecular dynamic simulations in order to simulate the forces that exist in real proteins. And using the molecular dynamic simulation, the predicted structure is predicted based on that in the environment of the simulated intermolecular and intramolecular forces, what is the structure that will adopt the lowest energy conformation. So using molecular dynamics methods, the ab initio prediction is done. So the advantage is that it works for novel folds. So knowledge-based method is based on known structure function relationships. But if you want a completely new structure, then this ab initio prediction is important. The problem is that it can only be applied to short sequences because long sequences take too much time to compute than is practically possible. There are several softwares for this ab initio protein modeling. The first one that we will discuss is called ITASR. The full form is Iterative Threading Assembly Refinement Structure Prediction Server. So the first step is called threading. It refers to a bioinformatics procedure for identifying template proteins from solved structure databases which have a similar structure or similar structural motif as the query sequence. So basically, suppose we have a sequence predicted for our protein. So threading refers to the, so there is a information, wealth of information that coordinates different structures to different sequences. So threading indicates that for our sequence, what is the structure that it will adopt based on the previously known knowledge of structure and sequence. Then comes structural assembly. Here, continuous fragments in threading alignments are excised from the template structures and are used to assemble structure conformations of the sections that aligned well with the unaligned regions built by ab initio modeling. So suppose the threading is predicting that the initial part of the sequence will adopt a alpha helical conformation. Then the middle part of the sequence will adopt a beta sheet conformation. So it will put together all these predictions into an initial predicted model called the template. There can be certain parts of the sequence that do not correspond to any of the known structures, and that will be predicted based on ab initio model. So now that we have the template, the fragment assembly simulation is performed again, starting from the selected cluster centroids. What this means is that you have the template, but now it can be, it is again improved. So based on more of this structure function knowledge, the structured assembly is done to get a more refined template. So here, if you can see the original template, there were some uncertainties in the template. But if a more refined structure function correlation is done, such that we are choosing only those structures that correspond the most to our protein, we can get a much more refined template. The final stage is called the structure-based functional annotation. The function of the query protein is inferred by structurally matching the predicted 3D model against the proteins of known structure and function in the PDB. So the final stage is predicting the structure against all the known structure and functions that we know in the PDB. So whichever structure our predicted protein is closest to, it will be able to correlate and predict the structure of our predicted protein. So that is how iTASR works. There is another software which is known as Rosetta Structure Prediction Server. The Rosetta software suite includes algorithms for computational modeling and analysis of protein structures. A suite of molecular modeling and search tools of Rosetta is used, which can calculate the energy of a folded protein and search for the lowest energy sequence for a given structure or the lowest energy structure for a given sequence. So what this means is that you have a given sequence, it will calculate the lowest energy possible for that sequence and what is the structure of this lowest energy. Or if you have a given structure, it will calculate that what is the lowest energy sequence that will adopt that particular structure. Although each de novo protein designing project is different, they all follow the same basic strategy. First, decide on a desired class of structures, a platonic ideal of a shape. Then use Rosetta to design tens of thousands of potential backbone conformations to match that shape and refine that shape using sidechain residues and test the calculated sequences that will fold into that desired shape. Finally, synthesize the genes that will best express that particular structure and using the results of the initial analysis, predict a better structure and continue this Rosetta design process. So the results of the initial prediction will help improve the results of the next prediction and so on. There are other methods of protein design. One very interesting method is called DNA origami. DNA origami is the nanoscale folding of DNA to create arbitrary 2 and 3 dimensional shapes at the nanoscale. The specificity of the interactions between complementary base pairs make DNA a useful construction material through design of its base sequences. So basically, since DNA has very specific interactions, like nucleotide A and T make 2 double bonds and G and C make 3 double bonds. So since it has very specific interactions, it can be used as a construction material because it is easy to predict that what are the interactions it will make and consequently what are the shapes it will adopt. Inspired by DNA origami, in which DNA molecules are folded into nanostructures. So in DNA origami, you can see several structures has been built, like this smiley face, or this vase-like structure, or this cuboidal structure. They have all been designed based on DNA origami, which is basically you have a random DNA and certain small fragments of DNA called staples are annealed to the random DNA to enable it to fold in the orientation that we want. It is possible to predict this folding because of the specificity of interaction between the DNA complementary bases. So inspired by this DNA origami, Chen wanted to identify hydrogen bonding strategies that would allow him to design perfectly orthogonal protein pairs. That is, proteins that would interact with a specific partner only and not with other proteins. So like this concept of protein design using the idea of DNA origami is still pretty new, but it has potential because DNA origami has given quite good results. The second method is called a blob level protein design. So the basic idea is to combine protein units of defined function to engineer a fusion protein with a new function. So for example, if you have protein A with function A and protein B with function B, you can get a protein which incorporates the structural elements of both protein A and B to get a combination of the two functions A and B. This is often used in sensors, signal transduction components, transcription factors, therapeutics, etc. Now, we will look into directed evolution. Just give me 1 minute. Right. So natural evolution of enzymes has existed since the emergence of life on Earth. Genes have mutated and proteins have evolved to improve the fitness of an organism to tackle conditions in new environments. Directed evolution of enzymes and binding proteins is a man-made procedure based on molecular insights, which moves the evolution process into the laboratory and speeds it up. Directed evolution is an iterative procedure which involves the identification of a starting state protein, then diversification of its gene. So what it means is that you start with a protein. So you start with a wild type polymerase gene. Then random variants are created with mutagenesis. These new variants are expressed using recombinant DNA in E. coli. And then this DNA is multiplied in tiny PCR microreactors. And after this PCR is formed, the variant proteins with desired traits are expressed in E. coli, and high throughput screening and selection of the candidates with desired procedures is done. So basically, when you get a lot of the mutated proteins, a question is asked, do any of the mutated proteins meet the desired criteria or the function that we want? If the answer is yes, then we have an improved and evolved protein. If it is no, then the protein is just process is again restarted with the best candidate from the previous round. So again, another round of random mutation is done. And this entire process, this repeating process, is called directed evolution. So it is an iterative process which involves the identification of a starting state protein, then the diversification of its gene using random mutagenesis, then an expression and screening strategy to find the best candidate, then again another round of random mutagenesis or re-diversification and rescreening, and so on until a satisfactory performance level in terms of enzymatic activity, binding affinity, or specificity is reached. Directed evolution of enzymes tailor them to operate in new reaction conditions, optimize their catalytic activity towards new substrates, and make them catalyze new chemical reactions. There are several molecular insights that guide the design of a library. Now, it is not possible to randomize every position in an enzyme, the typical size of which is 200 to 300 amino acid residues or more. A wealth of studies makes it clear that mutations in and nearby the active site, as well as more distant substitutions on the enzyme substrate, may contribute to optimized catalytic activity. So basically, you can see that there are several, okay, I'll just go to the next point now. Stemmer introduces a DNA recombination strategy termed DNA shuffling to the evolution of enzymes. This was an efficient way to propagate beneficial mutations while increasing the size of a DNA library through random fragmentation and reassembly of genes. So DNA shuffling consists of 4 points. Random mutations are introduced in the gene for the enzyme that will be changed. So if you have the DNA, you can see these red bars as the random mutation. The second part is that the genes are inserted in bacteria, which use them as templates and produce these randomly mutated enzymes. The third step is that the changed enzyme is tested. Those that are most efficient at catalyzing the desired chemical reaction are selected and the others are discarded. The new random mutations are introduced in the gene for the selected enzyme. So you have narrowed down those enzymes which have positive mutations. That is, they are somewhat catalyzing the reaction that we want. Then again, another round of random mutations are introduced, and this process is continued till the desired level of catalytic activity that we want is obtained. Selection criteria and screening techniques. So one criteria is thermostability. So when the directed evolution of enzymes is used with an aim to increase that thermal stability, the evolutionary process may be set up as interleaved heat treatment and activity assays. Or alternatively, the activity assay may be performed at elevated temperatures. What this means is after the mutation is obtained, it can be checked that with the increase in temperature, you can see that the wild type has a melting temperature which is lower than the mutant. So this mutant has greater thermal stability. This mutant can be used and to undergo a further round of random mutagenesis to get even more thermal stability, and the process repeated till we get the desired level of thermostability. The next is improving or changing catalytic activity. It is possible to evolve enzymes to improve that activity under new conditions. It is also possible to change that catalytic activity towards new substrates. The next is improving or changing catalytic activity. It is possible to evolve enzymes to improve that activity under new conditions. It is also possible to change that catalytic activity towards new substrates and reactions. This is possible as long as the enzyme that is chosen as a starting point has at least some low level of activity for the intended reaction. That is, some catalytic promiscuity. That means, suppose you have an enzyme that is catalyzing one reaction shown by this red, and you want an enzyme that catalyzes a new reaction. So there has to be some overlap. So that the enzyme has to be flexible enough such that it catalyzes the new reaction, say to even a very low degree, because that low degree of catalysis can be improved through random mutagenesis till a high level of catalysis of the new reaction is obtained in the mutated enzyme. So this catalytic promiscuity is important for making a mutated enzyme with a different catalytic activity. If an enzyme has a low level of activity for an intended reaction, but much higher activity for the natural reaction, it may be fruitful to first lower the natural activity before starting directed evolution efforts towards the new intended reaction. Few applications of directed evolution is now discussed. A strength of directed evolution methodology is the ability to co-evolve enzymes in biosynthetic pathways. In one example, Arnold and co-workers evolved a multi-enzyme pathway for carotenoid production in E. coli. So what was basically done is that the initial enzyme pathway for the carotenoid production in E. coli was identified. And then using this directed evolution methodology, they evolved a pathway which can be catalyzed by multiple reactions. So the advantage of this is that the production scale can be increased a lot because multiple enzymes are now able to catalyze the carotenoid production, and it is not restricted to a single particular enzyme. Another application is in biofuels. So a leading candidate biofuel is 2-methyl-propan-1-ol or isobutanol, which can be produced using a biosynthetic pathway in recombinant E. coli. Now, two enzymes in the pathway require NADPH as a cofactor, but while glycolysis, the normal metabolism during growth of E. coli produces NADH. To resolve this problem, directed evolution was used to alter the codependence of the enzymes so they can rely instead on NADH, making the enzyme and organism suitable for biofuel production. So the mutated enzyme can now use NADH to carry out its growth and metabolic activities so that it can be used as a biofuel. There are other applications like formation of new chemical bonds. So carbon-silicon bonds are common in human-made chemicals, but they are absent in biology. Nature has not evolved any enzyme so far that can catalyze the formation of carbon-silicon bonds. But directed evolution can be used as a strategy to ensure that such chemistry invented by humans can also be conducted with the help of enzymes. So starting from cytochrome C from Rhodothermus marinus, an evolved enzyme is created that catalyzes silicon-carbon bond formation 40 times better than its static enzyme. So basically, this introduces this principle, where a new reaction, the formation of the carbon-silicon bond, is catalyzed by a mutated enzyme. Another function is the enantioselectivity of enzymes. Now, directed evolution is an efficient way to improve the enantioselectivity of enzymes, that is, enhancing their performance in asymmetric catalysis. The evolved enzymes are used in the production of chiral substances with high enantiomeric purity. What this has been used for is that using directed evolution on transaminases, S selectivity, which was 65% in the conversion of the ketone B-tetralone to the corresponding amine, was greatly enhanced to 94%. What this means is that initially in this reaction, conversion of the ketone to the amine, the percentage of the S isomer formed was 65%. But using the mutated enzyme obtained from directed evolution, now the conversion to the S isomer is 94%. So the selectivity for one particular enantiomer is greatly increased. Now, what is machine learning assisted protein engineering? So screening is the bottleneck and most resource intensive step for the majority of directed evolution efforts. That means the screening is the step that takes the most time and effort. To reduce the experimental effort associated with directed protein evolution and to explore the sequence space encoded by mutating multiple positions simultaneously, machine learning was incorporated into the directed evolution process. So, as you know, molecular dynamic simulations, which predict dynamic structural changes for protein variants, have been used to predict changes in structure and protein properties caused by mutations. But full simulations are very resource intensive and require hundreds of processor hours for each variant. So a mechanistic understanding of the reaction at hand and ideally a reference protein structure is required for that kind of in-depth molecular dynamic simulations. Machine learning, on the other hand, comprises a set of algorithms that make decisions based on data. It enables rapid prediction of new variants, whereas further directed evolution campaigns may involve months of intensive experimentation. For example, if you have, suppose this is the initial protein structure that you're starting with, and as you go towards the inner side of the contours, you are going to a more stable protein structure. So molecular dynamic simulation will screen in all possible directions, and it will only follow, like after screening, it will decide on the path to follow. Again, it will screen in all particular directions, then it will decide on the path to follow. And like that, it will eventually reach a more thermostable structure. But machine learning, on the other hand, it will already narrow down. So it will take kind of samples at different positions. So at each position, it will take one sample, and gathering the data from that sample, it will be able to concentrate the predicted protein structure in a very small space. And so it will be able to reach the predicted protein structure very rapidly because it is not sampling all possible conformations, but based on the data of a sample conformation at each point, it is then predicting its next move. So how the model is developed in machine learning. Similar to quantitative structured activity or property relationship approaches that evaluate sets of features to the potency of the response variable. That is, labels for classification or regression for detecting beneficial ligand-target interaction in drug design. Quantitative sequence activity models for proteins were adopted. So what this means is that initially structured activity or structure property approaches were taken, but now the sequence activity approach is gaining much more advantage. So consequently, this quantitative structure sequence activity approach was established for elucidating features that are amino acid descriptors for detecting beneficial amino acid patterns and protein variants inside a defined sequence space. So basically, this sequence activity relationship was used in order to make a kind of information well in which several information is deposited, that what are the features of different amino acids that make them have specific function when they come together in a particular protein structure. Now, this information is what is used in machine learning. So the ProSAR algorithm fits a linear function based on sequence activity relationships to classify substitutions and hence identify individual substitutions that are beneficial or detrimental by deducing the independent contributions from the single residues. So using the information that is available through the sequence activity relationships, what this algorithm will do is that it will classify different mutations according to whether the mutation will be beneficial or it will be detrimental or neutral. And it will do this by kind of summing up the individual contributions from each amino acid. These identified substitutions that are predicted to be beneficial can then be introduced or deleted if they are detrimental from the next round of evolution. This is a technique that has been used to improve the catalytic function by almost 4,000 times by introducing 35 substitutions in a target enzyme. Now we will learn about epistasis. Now, mutations can frequently have a propensity to weak or no or negative epistasis. So, epistasis is a phenomenon in genetics in which the effect of a gene mutation is dependent on the presence or absence of mutations in one or more genes, respectively termed modifier genes. Epistatic mutations, therefore, have different effects on their own than when they occur together. A strong negative epistasis for accumulating mutations can likely lead to each other's extinction. When facing complex epistatic landscapes, machine learning is a useful method to find functions that can describe such behavior. So in the middle, if we consider this as wild type green fluorescent protein, which fluoresces green. Now, suppose this innermost circle is a single mutation. Most of the single mutations maintain the green fluorescent property, but two particular mutations lead to the extinction of green fluorescent. These gray dots mean that these mutations are no longer green fluorescent. However, the second circle, which implies another round of mutations, what can happen is that, as you can just 1 second, that one green, a green fluorescent mutation can either extinguish and become non-green fluorescent, or it can remain green fluorescent. Similarly, as other rounds of mutations are carried out, an interesting thing is observed is that one mutation which has caused the extinction of green fluorescent, after undergoing another mutation, it becomes green fluorescent again. So that is positive epistasis, meaning due to the presence of the second mutation, the effect of the first mutation is deleted. So the initial green fluorescent protein, which had become non-green fluorescent, becomes green fluorescent again. So this is epistasis because the effect of a gene mutation is dependent on the presence or absence of mutations in other genes. So in order to predict this epistatic behavior, machine learning is a useful method. Now there is something called as ePro, or Iterative Protein Redesign and Optimization Suite of programs. So what ePro does is that it redesigns proteins to increase or give specificity to novel or native substrates and cofactors. This is done by repeatedly randomly perturbing the structure of the proteins along specifically designed positions and identifying the lowest energy combination of rotamers and determining whether the new design has a lower binding energy than the prior ones. ePro also offers an integrated environment for grafting a binding pocket into an existing protein scaffold, predicting an antibody's tertiary structure based on the sequence, enhancing enzyme activity, assessing the structure, and binding energetics for a specific mutant. So suppose you have you have designed different mutant proteins, but only one mutant fulfills the energy cutoff for the targeted ligand. So that is the ligand that will be used for the protein design. Another example of designing a bacterial channel protein with a specific pore size. So basically, Pore Designer is a software that was successfully developed. That is the ligand that will be used for the protein design. Another example of designing a bacterial channel protein with a specific pore size. So basically, Pore Designer is a software that was successfully developed to redesign bacterial channel protein to reduce its 1 nanometer pore size to any designed sub-nanometer dimension. Two mutations were predicted. One is changing aspartic acid to tryptophan, and another is changing serine to tryptophan. Tryptophan, being a much larger group and a hydrophobic group, makes the hole size smaller as well as more hydrophobic. So like this, different predictions can be made. There is another success story in the field of protein engineering. So basically, computer-aided design has been used to engineer complex properties of a highly ordered nanoprotein assembly. A protein cage, E. coli bacteriophedin, which naturally shows structural instability, and an incomplete self-assembly models by populating two oligomerization states, is the model study. Through computational analysis and comparison to its homologs, it has been found that this protein has a smaller than average dimeric interface on its twofold symmetry axis, mainly due to the existence of an interfacial water pocket centered on two water-bridged asparagine residues. So the water pocket is shown here between the two asparagine labeled N residues. Replacing these two asparagine residues with hydrophobic amino acids like tryptophan or leucine results in proteins that fold into alpha helical monomers and assemble into cages, as evidenced by circular dichroism and transmission electron microscope. Both thermal and chemical denaturation confirm that all redesigned proteins, in agreement with calculations, possess increased stability. So basically, the problem was with the two water molecules that are held between the two asparagine. Because of these water molecules, the symmetry was lost and it was not able to self-assemble properly. Now, if the asparagine are replaced by hydrophobic residues like tryptophan, phenylalanine, or leucine, you can see that the relative tendency of cage formation is quite high in phenylalanine. And thermostability wise also, it is the best compared to all the analogs. Because in the other two analogs, while thermostability might be there, the cage formation tendency was not that much. So clearly the asparagine to phenylalanine mutation is the best for both thermostability and formation of the cage. Now there is an Institute for Protein Design, or IPD, which has 5 main goals. Firstly, it aims to build better vaccines for pathogens like the COVID virus. Second, it wants to expand beyond nature's alphabet of 20 amino acids. That is, it wants to make unnatural amino acids or non-canonical amino acids accessible for protein engineering. The third goal is that it wants to create advanced protein containers for targeted drug delivery. The fourth goal is that it wants to make smart proteins that can identify cancerous or otherwise unhealthy cells so that a targeted therapeutic approach can be undertaken in which only the cancerous or unhealthy cells are attacked. And finally, it wants to make next generation nanomolecules for use in solar energy capture and nanofabrication. There are several advancements in the field of protein engineering. One is ultra-potent vaccines. So researchers from IPD have created novel immunogens against the respiratory virus RSV, which is particularly lethal for infants and elderly. This technology was also used to create the ultra-potent COVID-19 vaccines that in animal testing found efficiency of more than 10 times than the antigen from the mRNA vaccines. Then there is cancer immunotherapy. In 2019, IPD created compact proteins that stimulate the same receptors as IL-2, which is a powerful therapeutic drug. But unwanted off-target receptor interactions are absent. So the tumor cells, which bind to the T cells to inactivate them, immunotherapy is blocking the tumor cells from deactivating the T cells, and thus the immune response is not suppressed. Another advancement is the rapid antiviral. In 2011, IPD designed synthetic mini-proteins that target conserved surfaces on influenza hemagglutinin from the 1918 H1N1 pandemic flu virus. Researchers from IPD have shown that these same flu binders can be used as diagnostic reagents in low-cost diagnostic test strips, providing improved performance as an influenza assay compared to a traditional antibody-based capture system. So what these rapid antivirals will do is that they can function in several ways. They can inhibit the fusion of the virus to the cell wall, which eventually allows the viral DNA to enter the cell. It can block the receptors on the cell wall, which does not allow the virus to attach at all. It can have protease inhibitors, which breaks down the viral proteins. Or it can inactivate the virus by blocking the virus's active site or changing the environmental condition or even destroying the viral structure completely. Then there is immune silencing. In 2014, IPD scientists were the first to demonstrate that computational design can be used to eliminate T cell epitopes from proteins by altering the amino acid sequence without affecting structure or function. The immune silencing approach enabled collaborators at the National Cancer Institute to improve an immunotoxin used to treat cancer. The new designed immunotoxins maintained good activity, stability, and anti-tumor activity. So what this immune silencing did is that it eliminated an immunotoxin used to treat cancer. The new designed immunotoxins maintained good activity, stability, and anti-tumor activity. So what this immune silencing did is that it eliminates the receptors in proteins which allow the T cells to bind, and it can do this without affecting the other useful structure or function of the protein. Now we'll look briefly into the tail of heme enzymes. So heme protein was viewed as a self-assembling, fully DNA-encoded catalyst whose electronic and steric properties can be tuned by evolution. It is an iron complex that can be modified simply by mutation of the DNA to make it capable of all sorts of interesting chemistry. Exploring the engineered enzyme catalysis towards transforming new substrates, especially abiotic reactions, opens up a new world of possibility. So there are several natural reactions that the heme enzyme can catalyze, like from CH to COH, or C double bond to epoxide, or RCOH to RH plus CO2. But now there are several non-natural reactions which it has enabled, like from C double bond C to cyclopropanes, or CH amination, or carbene XH insertions. So there is a lot of possibility for the non-natural reactions that this directed evolution of heme enzymes can catalyze. A single biocatalytic step at room temperature in aqueous solution replaces 5 or 6 chemical steps, often involving a precious metal, and provides an environmentally friendly way to make drugs. Now, there are new approaches and future directions for this protein engineering. Cadet et al. Came up with a supervised learning of enantioselective enzyme sequences and activity of n individual point mutations to predict the activity of all combinations 2 to the power n of these point mutations. This method involves numerically encoding the sequences. So if you have sequences, there is a numerical code for each amino acid, and then using Fourier transform to transform the amino acid sequence into a certain spectrum. Then the least square method is used to predict the activity of a mutant, which is a combination of multiple point mutations fed in the learning step. The correlation coefficient between 28 mutants validated experimentally revealed a good agreement. So what this means is that each of the mutants are first numerically coded and then Fourier transformed into a spectra. Then the protein activity relationship is determined. Using this information, an information bank is kind of made, and machine learning can be used to predict the activity of a new mutant. It was seen that when the data from 28 mutants was input into the machine, it was able to predict the activity of the new mutant with an agreement of 0.81, where 0.1 indicates perfect agreement. So clearly, if more information is fed into the machine, the agreement between the experiment and the prediction can be improved even closer to 1. Now that we have gone through the lectures of week 11, we'll quickly go through some sample questions. So the first question asks, what does Ramachandran plot show? So the correct answer is D. So Ramachandran plot shows the frequency of phi values of residues. Psi is plotted in the y-axis, phi is plotted in the x-axis. And it shows that which of the phi values are allowed and which regions correspond to specific secondary structures like antiparallel beta sheet or alpha helix or 310 helix. And these bold regions are fully allowed, the dotted regions are partially allowed, and the white regions are completely disallowed. The second question asks, which factors contribute to the stability of protein structure? So the answer is all of them. Because if you look at the different factors, you can see hydrophobicity contributes because there can be hydrophobic interactions between hydrophobic amino acids like leucine, valine, isoleucine, which basically have a hydrophobic side chain. So there can be van der Waals interactions or hydrophobic interactions between these amino acids. Electrostatic interactions can occur between positively and negatively charged side chains, like that between acids and bases. So that forms electrostatic interactions. Disulfide bridges can occur between two cysteine residues in which the SH hydrogen is removed and SS gets a single bond. So the correct answer is D, or all of them. The third question is that which of the following is not a component of PCR reaction? So the correct answer is D. So if you look at the components of the PCR reaction, the template DNA is required in order to build the new strand of DNA. The primer is required because DNA polymerase requires a 3' OH in order to extend the DNA. So it is only when we have a primer that the rest of the steps can take place. And finally, so we require the template DNA, which is the initial template, the flanking primer, and the dNTP, which is required for extension. So DNA polymerase in the extension step uses dNTPs to add on to the template strand. And so all ABC is required. So what is not required is D or DNase, because DNase helps in breaking the DNA, not multiplying. Then question 4 is the rational design of new protein molecules to design novel activity, behavior, or purpose And to advance basic understanding of protein function is called as what? So the correct answer is protein design. So as you can see, protein design has three major types, which is directed evolution, rational design, and combined methods. And we have discussed all this in this presentation. And protein design clearly is used to design new protein molecules with new novel activity, behavior, and purpose. And designing of these new protein molecules can give us a better idea of protein structure function relationships. So the correct answer is A. The next question is, what is de novo in protein engineering? So the correct answer is B. So the de novo approach is that we start from scratch where no knowledge of prior sequence is used. And sorry, we start from scratch. So basically, backbone building is done. And what this happens is that uh, what are idealized secondary structures of different sequences? Either that, so what are suppose the initial part 10 amino acids give rise to an alpha helix, the next 20 amino acids give rise to a beta sheet. So like that, the different amino acids that form the different structures are put together to have a reconstructed fold, or the fragments from different proteins can also be put together to get the reconstructed fold. Then using this information, a sequence is designed and optimized to have the lowest energy. And then experiments are used to find whether our predicted sequence has the required function. So de novo protein engineering is designing this new sequence based on different prior sequences. So the correct answer is B. The next question is, which of the following is the protein structure prediction method? So the correct answer is B, or homology modeling. So what happens in homology modeling is that homologous protein structure is identified. That is the structure that is homologous to the one that we want. Then the query sequence is aligned with the template sequence. And this is a very important step because this is what will ensure that the query sequence has a homologous structure to that structure that we want. Then a model is built for the query sequence using different structure prediction methods. So from the query sequence, we built the predicted structure. Then the model is evaluated and refined, and this can be repeated till we get the structure to be closest to that of our desired structures. So the protein structure prediction method is B, or homology model. Question 7 asks that ITASR structure prediction is based on what? So the correct answer is C. So what is threading? It is a bioinformatics procedure for identifying template proteins from solved structure databases, which have a similar structure or structural motif as the query sequence. So in ITASR, multiple threading approach is done. That is, if our protein is made up of structure fragments from multiple different structures, then it is used to match the specific structure to the that specific portion of the known structure, and that is the multiple threading approach. So the correct answer is C. D asks, what is silent mutation? So the correct answer is B. That is, a silent mutation is when the change in nucleotide sequence has no consequence for an amino acid in protein sequence. For example, if the initial sequence was TTC, which coded for lysine, and the mutated is TTT, where C is converted to T, and it still codes for lysine, then it is called a silent mutation, because the change in the nucleotide sequence has no consequence on the amino acid sequence. 9 asks, change in one nucleotide, like A to C, in the gene sequence of an enzyme is known as what? So the correct answer is substitution. So if you have a wild type of CTC nucleotide sequence and you mutate the sequence to get CAC, where the T is changed to an A, this is called substitution of one nucleotide, and it can cause change of an entire amino acid. Like initially CTC was coding for glutamic acid, and now it is coding for valine. Question 10 asks, epistasis is a phenomenon observed in the correct answer is B, genetics. So if we know about genetics, we can see that in epistasis, what happens is that if we see this, the coat color of mice, if there are two small c, the mice will be an albino no matter what the B pigment is. So if you can see these four white mice where it has small c, so no matter what the capital B denotes, that the mouse is black or brown, it does not matter, because the small cc gene is masking the effect of the other gene. So epistasis is a phenomenon in B, that is genetics. Question 11 asks, what are the requirements for directed mutagenesis? So the answer is C. That is, we need the cloned DNA of interest so that we can insert it into the plasmid that we want. And we need an expression system. That is, the plasmid needs to be inserted into the E. coli or whatever unicellular bacteria we are using so that it can multiply and make more of the protein. So the correct answer is C, that is both A and B. The final question for today is that what is the iterative protein redesign and optimization, or EPRO method? So the correct answer is C. That is, it redesigns proteins to increase or give specificity to native or novel substrates and cofactors. That is, native proteins can be designed in which the binding energy is improved, or even it is made more specific to novel cofactors or substrates. So the correct answer is C. That is the end of the discussion for today. So please contact me if you have any doubts, and I'll meet you on this coming Monday for the 12th live session.